Hello, everyone, and welcome back to Brushing Up on Quality 2.0 virtual conference hosted by Beyond Clean and sponsored by 3M, a full day dedicated to exploring dental reprocessing and infection control in the dental setting. I'm Brett Norton from Beyond Clean, and I just want to take a moment to thank you all for joining us today. For those of you anxiously awaiting your CE credits, after reviewing all of today's sessions, you'll have access to the full conference survey and will be able to download your CE certificate. If you need dental CEs, please be sure to check that box on the conference survey after the conference, and an additional dental CE certificate will be emailed to you. If you missed our earlier introduction, Dr. Nita Mazurat is a developer of new infection prevention and control guidelines for the Manitoba Dental Association. She is also a member of the Canadian Standards Association Technical Committee, where she is responsible for development and revision of Canadian medical devices reprocessing. Dr. Nita is a passionate educator, and she continues to write and lecture on the current topics in infection prevention and control and reprocessing for oral healthcare professionals. Have you ever wondered why testing for sterility assurance is critical during instrument reprocessing? Dr. Nita is here to kick off part 2 of her Dental Office Success Series to discuss the role testing and monitoring play for sterility assurance at critical performance points during instrument reprocessing. You'll be glad you tuned into this session, and without further ado, let's welcome Dr. Mazurat.
Hi, I'm Dr. Nita Mazurat. Welcome to part 2 of Setting Up Your Dental Office for Success, Sterility Assurance, Testing, and Monitoring. My disclaimer stays the same. The content of this webinar is based on current information, and I beg you not to rely only on it in isolation to make any specific decisions. My disclosure is that I am a very grateful educational consultant for 3M. This presentation has been designed to outline the role of testing at critical performance points during reprocessing, to describe qualification of sterilizers, products, and loads, to discuss the role of monitoring in sterility assurance, and to review or introduce new terms to dentistry that explain concepts essential to sterility assurance: validity, validation, process challenge device, qualification, requalification, and hopefully even some other terms that I have not included on this list that you can become comfortable with and start using in everyday practice. As a summary from our first presentation, sterility assurance is that integrated system of tests, controls, and backup procedures intended to ensure that reprocessed medical devices are sterile when delivered for use. And that's our definition from the Canadian Standards Association Standard Z31418. We talked about how that is achieved, a couple of the criteria, how that's achieved in our first presentation, which will be the live presentation, and it was following established guidelines and standards that are tailored to your office. You need to have your own standard operating procedures tailored to your individual office. Testing and monitoring at key intervals to ensure optimal equipment function and people function. Strategic planning for processes to be in place when or if failures occur. And I think it's when, not if, because failures occur and you need to know how to deal with them. And documentation, of course, to demonstrate functionality and adherence to office standard operating procedures. Review of the standard operating procedures, which we talk about in presentation 1. They operationalize the national, state, or provincial professional standards or laws to the individual office. You have to do that. The development of the standard operating procedures includes the development of an inventory of manufacturer's instructions for use, for cleaning, functionality, lubrication, sterilization parameters for all the instruments and devices in the office. This forms the basis, the groundwork for the complete picture of reprocessing tailored to the individual office. And you'll notice that I'm using the word reprocessing, not sterilization, because reprocessing is much more than just sterilization. And we're going to very briefly talk about existing design and how it requires offices to adapt to the challenges of their reality to the required standards. The role of testing. Testing at critical intervals for sterilization confirms that the process is implemented and successful as intended and planned. As a clinical dentist, for me to, although perhaps some of my endos looked like I hadn't done any radiographs or used an apex locator to determine the working length, well, with this, we need to use tests or diagnostic criteria just as we did when we are doing any other clinical procedures. We need to make sure that they're successful. You don't just do something and, well, for example, a crown prep. You can do the crown prep and then the crown comes back. You don't just cement the crown. You need to make sure that your margins are right, the occlusion is right, everything's fine. And it's the same with sterilization. It's not just pushing a button and hoping that everything will be fine. We need to make sure that we test it first to make sure that it's fine. So at key intervals to ensure that the process is performing as intended. In other words, we're going to have an endpoint of sterility so that we have devices, instruments and devices that are safe for patient use. The key part of this is that the physical state of sterility is difficult to prove. It's a state of nothing. It's a state where all microorganisms have been destroyed. So as soon as you touch something, of course, it's there. We were depositing microorganisms again. So it's very difficult to prove, especially clinically. So we have to accept the state of sterility has occurred because all the processes that result in sterility are in place and they've been correctly performed. Testing is performed on equipment and actual products or packages before the reprocessing for patient care begins. And this is what's termed qualification. And then afterwards, we continually monitor to make sure that everything is in place. So which comes first, bird or the egg? My granddaughter touching that sweet little duckling, or was the egg first? Well, obviously the egg was first, and there's no question here, much less philosophical and simpler in reprocessing. The equipment's installed, checked first to ensure that it's working the way that it was intended to work. But we have to do that. And I think a lot of times we take that for granted. So installation equipment is installed by the manufacturer. The manufacturers first provide us with the required information for the space for the equipment, including level countertops, the utilities, if specific electricals required, for example. And then the manufacturers themselves install the equipment and calibrate it. The combination of both of these requirements is, and I've couched it by saying sometimes referred to as installation qualification. In Canada, there's no question. We call it installation qualification, or IQ. Your office will receive documentation that this has been performed. Do not throw that out. You keep that, and you keep it forever. Qualification, qualified and qualification. Qualified means that the equipment or the process is demonstrated to be working the way that it was intended to work. Describes the equipment. And that was done by the technician who does the qualification. Means that a trained person has qualified the equipment. Describes the process. So the manufacturer ensures that the equipment is is installed correctly, and then that it's working correctly. They ensure that it works the way that the manufacturer intended it to work by calibrating all the gauges, that safety features and alarms are working, to warn you, for example, that you're short of water or that the electricity has gone off. So it's to warn the user when utilities or sensors are not working correctly. And it ensures that the equipment consistently meets the criteria that's required in your office for time, temperature, and pressure, because that's what sterilization requires. This is performed using a spore test in a process challenge device or a PCD, and I'll be talking, please don't worry about that yet. I'm going to be describing in detail how to develop your own in-office process challenge device, and when appropriate, a Bowie-Dick air removal test is performed, and we'll talk about that as well. This process is, and I know I couched it saying sometimes it's called operational qualification, or OQ. So the tests. I'm going to turn this around a little bit and say that because we're using, I'm showing 3 consecutive tests all the time. At the bottom of the page, I've got 3 consecutive successful tests have been deemed as an appropriate number to demonstrate that the equipment is working optimally. This is standard. This is just industry standard. That we use those 3 consecutive successful tests, no matter what we're testing. So if you're doing a Bowie-Dick or a type 2 test, it's run in an empty chamber of a pre-vac sterilizer. If you've got a pre-vac sterilizer, it is to ensure that 3 consecutive successful tests are completed, or 3 consecutive spore tests appropriate to the sterilizer type, whether you're using gravity or dynamic, in a process challenge device will be run for each cycle at the lowest time and or temperature to ensure 3 consecutive successful tests in a fully loaded tabletop sterilizer. Air removal, air removal is critical for sterilizer function. Only pre-vacuum sterilizers require the use of a type 2 air removal test because air always acts as an insulating layer and it prevents steam from contacting all surfaces of the devices being sterilized. And for sterilizers to be convenient for clinical use, the temperature and pressure reached by the steam that's produced has to be achieved quickly and consistently. And in order to have this happen, air has to be removed from the chamber, including the contents of your packages during the conditioning phases of the sterilization cycle. Air is removed. How the air is removed determines the classification of a sterilizer to gravity or dynamic. You need to know what kind of a sterilizer you have because the cycles are different, and when the manufacturer instructions for use talk about the parameters, you need to recognize what's gravity and what's dynamic. You need to know what kind of a sterilizer you have. Gravity sterilizers remove air passively. Steam is introduced, air is evacuated because air is heavier than steam. Air is forced downwards and out the drain. Unfortunately, air pockets, especially in a gravity sterilizer, can remain, especially in lumens, like handpieces, including dental hygiene handpieces and suctions. Dynamic sterilizers remove the air more effectively because a vacuum pump or a water ejector is used. Dynamic sterilizers are further divided into pre-vacuum and steam flush pressure pulse or SFPP sterilizers, depending on how the air is removed. I frequently have discussions with people who say that steam flush pressure pulse sterilizers are gravity sterilizers. They're not. Don't get caught into that discussion. They are dynamic sterilizers. I can't emphasize enough. You need to know what kind of a sterilizer you're using. Only pre-vacuum sterilizers, though, require the daily use of air removal tests. During conditioning, in a pre-vacuum sterilizer, during the removal of air phase, the sterilizer chamber is pressurized using a series of evacuations and steam injections, and during evacuations, the pressure in the chamber falls below atmospheric pressure. So, of course, the air, if there's a leak in the gasket around the door or any of the pipes or air, the air at atmospheric pressure would re-enter the sterilizer, and because of this reintroduced air, the sterilizer would be inefficient because you have air pockets again, and it wouldn't sterilize effectively or efficiently. So the air removal test is a type 2 chemical indicator, or we call it consistently a Bowie air removal test in Canada. So it's used in an empty sterilizer, preferably before the first load of the day, or it is tested at the same time every day in the location in the sterilizer according to the sterilizer manufacturer's instructions for use. And you need to have a pass. Okay, for our load test and release, let's talk about process challenge device. Because sterility is not measured easily or simply, we trust the process. It's determined because of sterility assurance. All the steps have been taken to ensure it has occurred. Part of that process is to ensure that spore tests that are performed to demonstrate sterility truly test or challenge the processes that result in sterility. A passed spore test provides the only true microbiological evidence that there is no growth of that microorganism that is very resistant to moist heat. The Geobacillus I knew it. I knew I shouldn't have even tried it. I won't. You can read them. A control that is from the same lot or batch of spore tests as the test spore test is incubated at the same time to demonstrate the spore test itself is viable. The conditions for killing the spores also need to be as difficult or challenging as possible. So the term challenging really does mean difficult. Difficult for steam penetration. It's the rationale for the use of the process challenge device, or PCD, to make conditions identical to real life at the worst case scenario. So if you placed a spore test simply into a peel pouch or naked into the sterilizer, which a lot of people unfortunately do, I find that it's similar as a prof. I felt that if I was giving you a test, a competency test, you had to pass that test. If I gave you the answers, the questions and the answers the day before, how would I be testing your competency? I'm not. So if I put the spore test into a simple peel pouch or just by itself into the sterilizer, I'm not challenging that spore test. I'm not making it difficult. I'm making it easy for it to pass. I don't want that. I need to make sure that my sterilizer is working properly. So you have to put that spore test into a package that makes it as hard as possible, but still possible to sterilize the spores, to know that the sterilizer is competent or working properly. So that's the PCD, or process challenge device. There are commercial process challenge devices available for hospital sterilizers, and there is disagreement in the industry about the validity of a commercial process challenge device, and I'm only going to say that. I'm not going to get into that disagreement in the industry. What I am doing is giving you a strategy provided by both AAMI, ST79, and Z314, the Canadian standard, that we can use in-house test package process challenge device, and they're developed unique to your office and unique to the challenges of the cycle that's being used, if you would like, based on the parameters required by the manufacturer for the devices in that specific load. So these are equivalent to the most challenged product, which means the instruments are the devices, and you'll notice that I'm using the term devices really covers, it's an umbrella. It covers the word instruments. So I would love to use the word devices all the time, but as a dentist, I have a tendency to use instruments. So forgive me for that constant back chameleon effect with instruments and devices. So product means instruments or device, plus their container, if they're in a container such as a cassette, and their protective packaging that's sterilized in your office, so whether it's peel pouch or wrap. Because every office is different, the standards are unable to provide actual devices used for making up your unique in-house test package PCD. You have to determine that within your own office as to what constitutes your unique in-house test package PCD for spore testing. Here's how. You choose the package in your office that is regularly sterilized and represents what you think is the greatest challenge to sterilization to copy as your in-office test package PCD. That can be the package that's the heaviest, such as surgical instruments, or has the most number of instruments, such as a restorative or an endo set, like whatever it is. Whatever you think is the one that you feel is the most challenging one to sterilize. So I said, for example, given a choice between a high-speed and low-speed handpiece in a peel pouch or a single high-speed in a cassette and wrapped, the more challenging is probably the wrapped cassette because of the cassette plus the wrap. I have no way of actually testing that other than using a BI, or actually testing it, yes, using a BI in both of them, making sure that both of them qualify. But it's almost a gut feeling as to which one you think, which one do you want to use as your most challenging. So the instruments in your test package need to be similar to that of what you've chosen as the most challenged, using lumens and solids, for example. So lumens, frequently more challenging, of course. And they're not necessarily, they don't have to be identical, but your numbers and your mass should be the same. Like, it doesn't have to be the exact same instrument, but if it's a restorative instrument, anything that you use as a hand instrument, you can replace those. I advocate that they're non-functional instruments that are broken, for example, no longer used, and now permanently reassigned to forming that package so that you always use the same package over and over. It just makes life much simpler for you, I think. And that reprocessing will, because by doing that, by repurposing those non-functional instruments, it maintains your current office inventory. I think dental offices often suffer from having a poor or a low inventory. So when we repurpose those non-functional instruments for this purpose, we're not further reducing our current inventory. Plus, remember that if you did use a functional set, that set would have to be reprocessed once you actually sterilized, and then had to check your CI, the chemical indicator, and remove the biological indicator, spore test, for incubation. So you'd have to reprocess that before you used it for patient care because it is no longer sterile. And always document. The composition of the set used as a PCD should always form part of the office SOPs. If the most challenged instruments or devices are normally sterilized in a cassette, the contents of the in-office test package PCD are also sterilized in a cassette. They'll be packaged the same way as the most challenged package, whether that's peel pouch or wrapped, however it is that you're doing it. The BI and a type 5 or 6 internal chemical indicator will be placed in the area that's most challenged within that package. And I've had discussion with colleagues from CSA, and they say, how can you possibly, it's like I'll argue about your determination. If you're constantly reprocessing, you have a real good gut feeling about where you think, or you're always a little uncomfortable. Is it because the instruments are packed really tightly there, or is it because you've got an instrument on the top part of the cassette and it's coming down and it's against the other ones? Like, wherever you feel is the most challenged, if you're able to place your BI and your type 5 there, that's the place to place it. Finally, the test package is placed in the most challenged area of the sterilizer according to the sterilizer manufacturer's instructions for use. So I just want to back up, do a little summary here, get my ducks in a row. Let's make sure, first of all, that the sterilizer has been installed properly on a leveled surface. Utilities required are in place. It's been calibrated. We've tested to make sure that we've got 3 consecutive successful air removal tests for pre-vacuum sterilizers, if that's the kind of a sterilizer you have, and then testing to ensure that the sterilization is occurring by testing spore tests in a PCD, and that 3 consecutive successful tests were performed. So your final step in preparation for the real thing is ensuring that the tailored packaging in your office results in sterile patient care items. Again, I think that we have a tendency to just do our packaging, put it in the sterilizer, let it run. When it's finished, and we don't think a lot about the fact that the way that we've conformed or placed our instruments into our packages, that is really going to result in successful sterilization. So that's called qualification and performance qualification of your packaging. So the parameters for the sterilization as validated by the manufacturers have to be known and followed, and that's why you need to have a device inventory, which is part of the discussion from presentation 1. Knowing your validated manufacturer's instructions for use assures you that the items that have been put together as product sets and sterilized in the correct cycle have all been validated for the same parameter requirements. And let's talk about validation and testing. Validated, I keep using the word validated. Validated is a word, please start using that the next time you go into the office. Validated manufacturer's instructions for use means the manufacturer has had a company or a division of their own company that tests the instructions that have validated or confirmed that these instructions are used as provided so you've got the instrument or device, you've got your manufacturer's instructions for use. It's known that if you follow those manufacturer's instructions for use exactly as required, that device will be clean and sterile, and most important, that they're now going to be safe for patient care. Oh, a little bit of magic. Sorry, I meant to remove that. That's awful. Performance testing or qualification. So every device that is reprocessed in the office is assigned to a product group characterized by similar attributes. So your restorative sets, your endo sets, surgical sets. But the other similar attribute is that they are processed at the same parameters. Chances are you're already using them that way in your office. They're based on how the operator likes to have them presented together for use, and but you need to make sure that their parameters for sterilization will include that. Your examples of product sets or families are devices in cassettes packaged in wrap, devices in cassettes packaged in peel pouches, devices in peel pouches as per manufacturer's recommendations for no more than the maximum number of devices, and they all have the same parameters for sterilization. Devices that need to be sterilized in extended cycles, such as rin holders, other plastics, and hygiene handpieces. Many of the hygiene handpieces for the ultrasonic scalers are run at longer time, and it's because they're lumens. So plastics are frequently longer or extended cycles. The handpieces are, some of our handpieces in dentistry, also extended cycles. Please check your manufacturer's instructions for use, and when you are sterilizing, you need to make sure that you follow those manufacturer's instructions for use. Performance testing steps. Out of each product group, choose the product that represents the greatest challenge at each set of test conditions. So the shortest time, the lowest temperature. So if you've got 132 times 6 and 132.4, then you'll do it at 132.4 because that is a much more challenged condition. where the instructions from the manufacturer state that a range is allowed. Clearly label your packages as test packages. Remember, we're doing performance testing here, performance qualification. Each representative product contains external chemical indicators, okay? Internal type 5 or 6 chemical indicators, and you can use more than one. If you've got a number of areas where you're uncomfortable, remember what I said about there being instruments on the top and on the bottom, and they're hitting each other during sterilization. Place your type 5 or 6 in that area. So because you're probably uncomfortable that, is steam actually penetrating that area? And BIs wherever you can as well. So you may have 3 or 4 BIs in those packages that you are performance testing. Other areas, those little areas in cassettes where we pack it contains other things, other instruments, devices that are not in the individual slots, and we have a tendency to overdo those areas, especially the long, narrow ones. At interfaces, where I said where they're clipped, so use multiple BIs in each representative product. Use them where you feel that there may be a problem. It's to make sure that steam penetration has occurred. Remember, we're testing to make sure that the cassette configuration that you've got is actually going to be effective, that they will sterilize. Test in a fully loaded sterilizer. Load according always to the manufacturer's instructions, and document your configurations as load configurations. Take pictures. That's the easiest way to do it, because this is going to be used for training. And you need to have 3 consecutive successful tests required for each appropriate cycle. At the end of the first testing cycle, internal chemical indicators are removed. Results are always documented, and only if they pass are spore tests removed for incubation with their and only if the chemical indicators pass, then you remove your spore tests for incubation with their controls. Contents are then quarantined as configured until your results are known. So, configuration, that means how you arrange your instruments or devices in the cassette, or the numbers of types of instruments that are in a peel pouch. It's important to do that for now. All the products are repackaged, and new chemical and biological indicators are again done. So you remember, because you're doing this three consecutive times, and you need to have passes for 3 consecutive successful tests. If any tests fail, products need to be reconfigured so that those areas, like where was the problem? So if you had multiple BIs in the cassettes, you should label them so that you know where they were. Otherwise, it's not useful for you. So 1, 2, 3 for the areas. And remember, you took pictures anyway, so that you'll be able to know, have those spore tests clearly labeled so that you know where they were in case of a failure, and then reconfigure and do it again. Loads are monitored, as with all released loads, based on past physical monitors, chemical indicators, and biological indicators. And it's time for some comic relief. Needless to say, this would probably not pass. Hopefully it doesn't look like anybody's that we've seen. And thank you to Dr. Chris Swayze from the Royal College of Dental Surgeons of Ontario for that wonderful picture. Reconfiguration means that the loading of the cassette or the pill pouch contents needs to be changed. Failure may be due to too many instruments in the cassette, too many instruments clipped into the upper cassette, more instruments or devices in the peel pouch than allowed by the manufacturer. Once the failed products are reconfigured and subsequently pass, the new configuration will constitute the way a package is configured. You have to document that so that it's used throughout, so that everyone knows, is trained in a standardized way in your office as to how to configure, or how the instruments are laid out in the cassettes and how the peel pouches are filled. If more than one new sterilizer is being qualified, then every sterilizer has to be tested, of course. Everyone has to be qualified. The product test sets are not used for patient treatment until they're reprocessed, using all the steps for reprocessing. When new packaging is introduced, or if individual packages are reconfigured by adding more devices, they need to be requalified. That means 3 consecutive tests. A successful test. When new load configurations are introduced, we take something away, we add a new type of instrument. Although rare for dentistry, it would apply due to introduction of change or use or change of type of cassettes, for example. They need to be requalified. New equipment has to be tested to ensure that it is working correctly at installation, known as qualification. Following failures and major repairs, equipment is tested again to ensure proper function, known as requalification, using 3 consecutive successful tests. Have to be consecutive and successful. I know I'm a broken record. There's a reason for that. I want that to stick in your head. Following installation, equipment is tested continually by monitoring. Now we're talking what you do every single day. Once the sterilizer is qualified, it's known to be working correctly, the way that the packages are configured and the load configurations are confirmed, the sterilizer and the individual products are regularly monitored for sterility assurance. This is what you're doing. Every load with physical parameters, packages with chemical indicators, the sterilizer with biological indicators. Physical monitoring for every load. The physical or mechanical monitors are the gauges or readouts, printouts, or electronic data recorders, and they confirm that the parameters of temperature, pressure, and time required for sterilization were reached. They have to be confirmed at the end of every load. Every time the sterilizer is run, and the loads are only released when it's confirmed that they have passed. Documentation with a signature on the load log is required to demonstrate that these were checked. If a sterilizer does not have a printout or an electronic data recorder to check it, then an observer, somebody has to stand there and check that they were reached during the cycle, and then we need to document that these conditions were met with their initials on the load log. Printouts are not kept because they fade, it requires storage, searching to find it. So instead, that information is documented in the load log, and it's documented essentially just with this signature that indeed it happened. Chemical indicators. There's 6 types of chemical indicators. They're all used for different purposes, and they're by type, showing there's really not a hierarchy. They're just chemical indicators. Type 1 is an external chemical indicator. Type 2 is the Bowie-Dick. Type 3, I don't think I'd ever seen one. It just monitors a single specific parameter. Type 4s are being phased out in Canada because they only monitor 2 specific parameters. We use type 5 and type 6 all the time, and in every type 5 or 6 are used in every single package that is placed into the sterilizer. Type 1s are your external indicator, unless there's an internal chemical indicator in a peel pouch and it's visible from the outside, but you don't position it just for that purpose. External indicators are used to ensure that the load has been processed and should be checked when you open the sterilizer, when you're removing the packages, when you're placing them into storage, and when you're transporting to patient care. It's just one of those things that you bang, 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 do. I get more calls from people who have said that. What do we do? What do we do? Somebody loaded the sterilizer, but they didn't start it. Somebody else came along thinking that it was finished, it was done, it was cool, and they unloaded and placed those packages into storage. They didn't do all those things. They didn't check the parameters. They didn't, when they opened the sterilizer, they didn't check the external chemical indicators. To me, the packages feel so different when they're sterile, so I don't understand how that's happened, but it does. Busy people are busy people, and it happens. Get yourself into the habit of checking all those 4 times. If an external indicator has not passed, please don't use those. It's not a question of please. Do not use those instruments. Troubleshoot to determine what's going on, what's happening. Internal chemical indicators, all devices that are reprocessed need to be packaged. All internal indicator or an internal indicator is placed in the most challenged or difficult to access area of every package, and that's hard, especially with a peel pouch because it falls to the bottom, but it's still hopefully between instruments or devices, and it's to determine that the steam has reached the most difficult to access area in that package. Internal chemical monitoring causes the most amount of confusion, the most amount of variation in requirements between the provincial or state dental standards, even in their most recently revised standards. In Canada, a type 5 or 6 internal chemical indicator is required in every single package. Spore testing. Biological indicators or spore tests are used for continued safe sterilizer operation. They're performed using a BI and a chemical indicator in a PCD, as we described earlier, and with the frequency required by the standards by your provincial or state licensure regulator. In Canada, as I said, it's done daily. In the United States, the AAMI ST79 standard is the use of BIs and CIs in PCDs weekly, but preferably every day that the sterilizer is in use. There are spore tests that can be incubated actually in 10 minutes, 20 minutes, 24 minutes, 1 hour, 2 hours, 24 hours. And the spore test and the incubator have to be compatible, and the spore test must be compatible with the type of sterilizer. The lower your device inventory, the better it is to use a BI with a shorter incubation time, because if there's a problem, you'll have fewer patients that you have to contact regarding a potential problem. A control spore test from the same batch or lot is, of course, incubated at the same time to ensure that the spore test is viable. And if any of the monitors fail, do not use the devices in that package or depending on the failure in that load. And you need to have written processes in place for your office. That's going to be the topic of our next presentation. So for a summary of what I just babbled about, we discussed what may be a lot of new information for many dental offices, how to qualify sterilizers, and the sterilization process. This is an important aspect of sterility assurance. I keep calling it insurance. Sorry. It is a type of insurance, but it's assurance, and it's an essential building block to ensure that your equipment and the configurations of your individual packages and loads can provide your office with consistently safe, sterile devices for safe patient care. The next presentation will focus on the last aspects of sterility assurance, which is planning for failure and documentation. I'm Dr. Nita Mazurat for Sterility Assurance. Thank you.
All right. Thank you for that. That was great. And I wanted to welcome back Dr. Mazurat once again. Thanks for joining us this afternoon. How is it up in Canada this afternoon?
It's gorgeous. Absolute gorgeous afternoon. Absolute.
Great. Well, I know that we had this session recorded for some reasons, but I wanted to get your opinion, or wanted to see if you wanted to shed any light on the presentation itself, on the importance of testing and monitoring, and then we can get into some more in-depth questions, and maybe just to share with our audience live your overall view on the topic itself.
Yeah, first of all, thank you for the opportunity for pre-recording. I almost wish I hadn't. My voice is awful. I couldn't stand listening to myself, so I apologize to everyone. I don't know what that was all about. And also, I used to say I can't walk and chew gum at the same time. Now I say I can't pre-record and say Bacillus thermophilus at the same time. Like, what was that? A reprocessing person who can't say the most important ones. It's like, wow. Okay. This is tough. This is stuff that we haven't heard about too much in dentistry, called IQ, OQ, PQ, installation qualification, operational qualification, and performance qualification. it's just not something that we ever talk about. So I've got there have been some questions here about who's supposed to do this stuff. Do you want to start there? It's kind of the third question that
I can
was asked, but do you want to start there? Okay.
Sure, absolutely. I can go ahead and read that off. And
sure.
When it comes to new equipment, who is responsible for new equipment testing? I think there really needs to be a full range when it comes to the new equipment. There needs to be the clinician and whoever's operating the equipment and using the equipment on a daily basis. They have to familiarize themselves. But when it comes to the introduction of new equipment, is it the manufacturer that is responsible? Is it the sales rep or the dental hygienist or the front office assistant? How about you shed some light on that?
Sure. Absolutely. It's the manufacturer and the manufacturer's representative. So if it's the salesperson and they're qualified to be able to they're trained to be able to do the qualification, then they can do it. But it's not up to the individual office to do that, from what I understand and from what's done in Canada. So the manufacturer makes sure that they provide what is required for installation, so flat surface, the utilities, and so on. And then they're supposed to bring it in, and they help with making sure that the bells and whistles are right. So that's installation qualification, operational qualification, and then performance should be done in a combination with the office, because the rep doesn't know what you're using. So you have to provide the way you're going to be putting your instruments in the cassettes or into the peel pouches so that you can qualify it with them. And it requires their knowledge as to, it's good to have a second eye or an objective eye to say, that looks like it's a more challenging position. Let's put a PI and CI in there. There's another area. When we're doing this, it's not just one little BI in that cassette. You might have up to 3-4 BIs in that cassette, just to make sure that every area is fine. And that this is under the most stringent conditions, so that makes sure that when you're doing your regular sterilization day by day, that you can trust it. Does that answer your question?
Yeah, I think it's the saying, "Trust the process," and also,
absolutely,
when you're talking in terms of space constraints and different ways of operating, because there's bigger offices, there's busier offices, you've got more space some have more space for reprocessing, some have very little. And those are, I think, some very important points to take away, that it has to be geared toward the needs of the dental office itself, and making sure that you're showing them the new equipment testing based on their process. And the one question I would have, though, is, say if you are the manufacturer and you have a question mark on their process, is it your job to say, listen, that's not the right way. This is something I would suggest that you change, to maybe alter your process so it's a much more efficient way to go about your sterilization? Or how would you recommend that?
That sounds very similar to what you and Peggy were talking about.
Yep.
Like when you can see something, and obviously you're traumatized by this. This must happen to you a lot, or something.
Well, I think it's a very interesting situation, because there are so many times when you can see something and do nothing. But I really am a proponent of seeing something and doing something about it if you feel that it's not the right process.
Well,
so
you're educating. You're educating. And Peggy said it so well, that there's a fine line between ethics now. And, if you didn't point it out and you know better, then it's up to the office then to accept what you've suggested or tried to teach. But if you didn't point it out, you're just adding to the problem. And they may at some point say, hey, they never told me that. So yeah, I think it's up to, I mean, for all of us, if we see something that's not right, we need to say it. We can't be judgmental about it. You just say the standards say, or this is the way that I've been educated to do it, and you can show them or tell them, provide them with the evidence. And then what they do is up to them. But hopefully they would be accepting of that.
Right. Yep. So kind of talking a little bit more on the maintenance side, how often would you suggest maintaining, equipment as far as is it based on use, or, we had talked about earlier standard operating procedures, right? So regardless of if it's used X amount of times per week, should you just have a standard in process so it's just continuous and the maintenance is there, and so you know that everything is working correctly?
You're talking about actual maintenance of this, cleaning and maintaining equipment? Is that what you're saying?
Yeah, of the equipment that's, that you're sterilizing your instruments with. And, obviously the testing and monitoring goes into that as well. But,
little different. The actual maintenance is generally provided in the manufacturer's instructions for use.
Right.
And not only do we follow that, but you need to document that. Right. Just as people have said before me today, document. Show it, demonstrate. And that becomes a practice builder. It's also when you sell your practice, you can demonstrate due diligence, that you've been careful about how you look after that equipment.
Right.
Did that answer your question?
Yes.
Yes.
Okay, so when you have a digital printer on your sterilizer, how can you initial that so that it meets the sterilizer parameters? Do we print them off after each load? And that's a good question from one of our audience members.
Yes, thank you that means I've actually got audience members out there. That's really exciting on a Friday afternoon.
Quite a few, actually,
I'm grateful. You your digital recorder, and then you initial it live, like it's hard copy on your load log. And I'll be speaking about load logs and documentation next. So that's how it's done. You can print it off, but wow, then you're going to have reams and reams and reams. We just can't do that because we just can't keep stuff forever and ever. You can if you want, but it's really difficult. It's more important that you state in your standard operating procedures what you're going to do, how you're going to do it, and then document that you've done it. And it's just consistent, and that's how it's done. That's sterility assurance.
Yeah, and I think when it comes to, say, an OSHA visit, if they're asking for documentation, if you have it and you're very diligent about doing that, you can just pull it out and say, hey, this is what we've been doing, and away you go.
It's an expectation. Yeah, it's an expectation in your office.
Yep.
Absolutely.
And it should be.
Yeah.
So you talked about the types of indicators and, Bowie Dick and variety of others. Are there any types that you would recommend using above all others? Or can you explain some of the main differences between the indicators?
Well, a type 2 Bowie Dick is only used in a pre-vac, and it's used daily. And that's to make sure that you're removing all the air. Okay. So that's type 2. In Canada, we have to use a type 5 or type 6 in every single package. That's to make sure that the steam has actually entered that package. It may not if the packages are very tightly packed together or against the sides of the sterilizer. In other words, the loading has not been done correctly. Could also sometimes be packaging, could also sometimes be the lot numbers. You may have a type 5 or type 6 that's not working properly. So it'll give us a clue that there's a problem with that. Again, it's going to be different in the United States. Right. It's different state to state. So right. I'm a little leery about saying that. All I can say is what Z314 says.
Right. Yeah. No, that's a good answer. So, you brought up the Bowie Dick, the type 2 test that tests for air. And this may be going down a little bit of a rabbit hole. And, I think, and this goes for also on the medical side of things as well, with all the advancement in medical instrumentation. There are more, very, very fine, delicate instruments that have very small lumens and very tiny channels. And, the lack of drying within these lumens can lead to real issues with sterilization. And so, this is a discussion that could probably go on for a long time, because drying is definitely becoming more of a focus now in the industry. But do you have suggested ways of drying the channels to where you can adequately test those and make sure that, they're testing safe, I guess?
Testing safe. Well, I golf with Michelle Alpha, who's the endoscope guru in North America. She recommends warm instrument air. We don't use a lot of instrument air in reprocessing areas in dentistry. I can see us starting to do that more. You should always, if you can actually see all the way through the lumen, it should be dried with instrument air, and then you should use a light. And I'm going to back up for a second. You always put something to catch any of the moisture on the distal end. Then you check it using a light, for example. I'd love to see very tiny lumens using borescopes for them. Mm-hmm. And now we're going to have dentists going, what are you doing to me? But think about it. Think about some of those really small surgical suctions that have a bend in them. How on earth can you test or can you visually inspect one of those? You cannot.
It's impossible.
So
yep.
It's impossible. So if you're going to continue using those, you're going to have to use something to help you, like a borescope, or use a disposable device, just as was indicated to us earlier today, to use a disposable adapter for HVEs and saliva ejectors, because it takes so long to clean those adapters, but it still has to be done and they should be sterilized. So same thing with these. If you can't do the cleaning properly, and part of the cleaning, part of the reprocessing is the inspection, the drying and the inspection. If you can't do that, then switch to a disposable device. And I apologize to generations to come, because those things are going to be in landfill forever. I just don't see another choice.
Yeah, I agree with you. And I think that is, as you said, it's on the endoscope side of things is becoming more of an emphasis, and also with robotics, because yes. you cannot see through those channels, and it's hard you can't determine, 5-6 inches down what is dry and what is not. And,
Clean.
The old way. Yeah, exactly.
Clean. Right. Dry alone is not a problem. Clean is the problem.
It's true. And, and then it's using the correct chemicals, the correct processes, and
brushes. Yep. Yeah, not just running water.
Right.
You have to use the right brush. It has to be the right kind, the right diameter, the right length. so that it does actually come out the distal end, just enough so that you can actually see, but follow manufacturer's instructions. And if you don't have them, get them.
Yep.
Ask for them. Validate.
Yeah, and you probably heard my talk with Peggy, is that if you don't have the instructions for use, you need to have them readily available, or you need to have a resource for them. So it's a very necessary tool in making sure that you're doing your job correctly. So,
yeah. Yes.
Well, I will let you sign off and get a quick breather for our final presentation, also featuring you. And just wanted to say thanks again for joining us. We're almost there, everybody, but thanks all of you out there for joining us throughout this day. It's been a great day. It's been some eye-opening new ways of thinking as far as educating our professionals in the industry, and we can't thank you enough for joining us. So stick around. We've got one more in 15 minutes, and we'll be right back with you. Thanks again.